Thanks, Simon, and thanks, Richard, again for the great invitation. And I have to say, this is one of the best meetings I think I've been to. The quality of the talks and the individuals, of course, have been absolutely fantastic, so thank you to all. I have changed my title a little bit to make it a little bit more palatable. I believe the goal was also to show some of the technologies that we're using. So to remind you, I did speak a few days ago on some of the studies we were doing and how we got to the point of doing a phase 3 trial, which was approved by the FDA in the US and Health Canada, as well as some regulatory bodies in Africa and in South America. We're just waiting on Europe now. So in general, I'll just talk a little bit about the workflow, how we got to where we are. We started off by doing a large-scale international study. We collected samples, blood samples, plasma samples, and we did proteomic analyses, and you've heard quite a bit about proteomic analyses as well as other omics at this meeting. Essentially, we used a technique called O-link, which we prefer. We feel it's one of the superior techniques, simply because every protein has to be bound by 2 antibodies, and each antibody is conjugated to an oligonucleotide, which are complementary, and if those antibodies bind close together, there's hybridization, and then that signal can be amplified. So it has incredible sensitivity as well as specificity. We then went on to identify differentially expressed proteins. And those differentially expressed proteins serve two purposes, actually. We can go down the biomarker route, where we look for diagnostic markers, prognostic markers, and treatment response. And then they can also be used to look for signaling pathways, which then could be used for drug repurposing. Anytime a pharmaceutical agent or pharmaceutical agency is now developing drugs, they are trying to co-develop biomarkers. So they really go together. The reason being, particularly if you're dealing with subjective information, is you really want to have a treatment response, something objective that you can measure. So the ideal scenario for any clinical trial is to have a biomarker and a drug which are somewhat married. Going back to some data that I showed just a couple of days ago, I'll just show this, this brief slide, which reflects our differentially expressed proteins between our long COVID positive patients on the left and our healthy controls. And on the right, you've got the long COVID patients versus the long COVID negative. The red, of course, reflects statistically significant upregulated proteins. The blue represents downregulated proteins, about an 80-20 split, which isn't uncommon in inflammatory conditions. And then when we compared the long COVID positives to long COVID negatives, we went down in the number of proteins, and in the disease state, those that were only seen were upregulated, and they still had strong statistical significance. They didn't have the strong fold change that we saw against healthy controls. But we took this data, and again, we're going to use it for 2 approaches. One is biomarker discovery, one is for drug discovery. So I'll just talk briefly about our approach to biomarker discovery, where we've had quite a bit of success. We use machine learning techniques, and so we'll take the entire set of data. We try to take the raw data so we have the biggest idea of what's going on. We split into training data set and testing data set, usually about a 70-30 split. And we do 2 machine learning techniques, a Bruta feature selection followed by a recursive feature elimination. The first approach, a Bruta feature selection, is relatively conservative, and essentially it removes all the proteins that are close to random noise. The second technique, recursive feature elimination, is a little bit more aggressive, and it essentially drops all the poor performers. In our hands with proteomic work and looking at a number of different diseases, typically with a Bruta from a large data set, we can get down to around 20 or 30 proteins. And then with a recursive feature elimination, we can take it down a little further and get down to 10 or less. The way I would describe the procedure, it's like having a tree. That tree has branches, and some of the branches are live, some of the branches are dead. And using these techniques, you essentially start off by pruning all the dead branches, leaving only the live ones, and then you look at the live ones and you think which ones are actually going to grow, and then you get rid of the other ones. So you're left with a tree which just has very meaningful branches. We then use a random forest, which is a set of decision trees, which is very helpful in determining feature importance. So in terms of ranking those biomarkers in terms of what's most important. We use all your traditional statistics, ROC curves, F1 scores, which incorporates recall and precision, sensitivity, specificity, and our feature importance. What we've been able to determine from this large data set in terms of biomarker discovery, we've been able to identify groupings of 4 to 8 proteins which have a ROC value of about 0.86 to 0.88. If you recall your ROC, how to interpret a ROC, anything greater than a 0.7 is considered good, anything greater than 0.8 is considered very good, and 0.9 is considered excellent in terms of diagnostic capability. We're sitting just below the excellent category. When we go into our PR curves, we sit around 0.99. So a PR, that would reflect the average precision for the number of recalls. Anything greater than 0.95 is considered good. 1 is considered excellent, so we're very close to being excellent in terms of precision and recall. Right now, we have signed a contract with Thermo Fisher that makes your multiplex immunoassays. We felt this was the best way to go to complement the study. For those of you who are not familiar with it, it uses bead technology. Those beads can be excited by various wavelengths, and they emit certain signals, and by that you can tell which capture antibody they're attached to, which then captures the analyte of interest, and then there's a fluorophore which you measure, which gets converted to a concentration. This typically takes about 2 hours in the lab to run a sample, and all of the proteins can be run at once. We have another technology. I'm just going to show you briefly. It's not ready for prime time. It's something we've been working in my lab for about the past 5 years. We're hoping to have this to fruition at some point. This is a point-of-care immunoassay which uses lateral flow technology. This is we would use a capillary sample where you lance your figure, you use the pipette, you draw 40 microliters, you drop it into a little capsule which has all of your conjugates. And we use something called nanodots. Nanodots fluoresce, and nanodots are about 20 times more sensitive than your standard colloidal gold. We can use your standard nitrocellulose paper, drop 5 drops in each of the samples, they run down it, and it's a semi-quantitative test. I'm just not sure with the separation that we have in long COVID right now that a semi-quantitative test would be accurate enough. And then we use a $2 flashlight, which has the correct excitation to measure the actual proteins on the nitrocellulose. So that's something that may be coming down the pipe in the future. Now, the other way we used our differentially expressed proteins was to determine signaling pathways, and if you were here a few days ago, you saw the different approaches we used. In this particular case, we tightened up the data by doing both normalization and batch corrections, and then we fell back on our hallmark pathway techniques, and we found 13 pathways that were not only statistically significant, but those 13 pathways correlated with all of the different geographical areas that were involved in the study, and that's shown in the chord diagram in B. And then in C, those pathways also were strongly associated with all of the symptom clusters. So they seem to be important for pretty much all patients from all areas of the world. The darkness of the line reflects the biological impact, but all of those lines are statistically significant. There's, of course, variability from site to site, and there's variability from symptom cluster to symptom cluster, but those 13 pathways are relevant for all patients in all areas. So what do we do with this data now to get to drugs? Well, first of all, we have to have some very simple assumptions. So the protein-disease relationship is important, and in a very simple sense, you have a compound that has a target, which is your protein or proteins, and those proteins are associated with disease, and you can take the inverse look at this, that the disease is implicated by the protein, and it's a target of the compound. So we go into it with that simple philosophy, and we get into our drug repurposing. And if you recall from a few days ago, we did it independently with a group from UPenn, and we came up with the same drugs. They were in different orders in terms of our ranking, but both drugs came up from 2 groups independently from the data. Typically, we would take our proteomic and our bioinformatic data as well as data from the approved drug databases, and that goes into our machine learning engines. So we look at pattern recognition. What we're doing there is we're looking for the molecular signature of the actual proteins themselves, and we're also looking at the drug effects. We then do network analyses to connect the two, and then we get into vector mathematics to make some predictions, just to increase the idea there. So we have our raw biological data, which goes into, it gets converted to a numerical vector in a very high dimensional space. That goes into our machine learning algorithms. and we can create vector space visualization. So this is a transformed space where you can have your drugs, you can have your diseases, and depending on how far that separation is between them, you start to build relationships. We can then get into some mathematical models using cosine similarities and other things. And essentially, what we're looking is for the distance between the drug and the target and trying to marry the two together. And we can create rankings based on the distance between the two. Something we're trying to work on now, we're not there yet. Once we have our candidate drugs for a particular disease, what we want to do now is integrate other information that's important for the patient. So not just the actual coming together of the drug and the disease, but we want to work in those biological and pharmacological aspects. So things like absorption, metabolism, excretion, volume of distribution, and most importantly, safety profiles. We're not that far yet, but that's coming down the pipeline as well. And I think in the future, this will be a very automated process to go through. What did we find? So we found the 13 pathways. Here I'm just illustrating some of the pathways. This is what I showed a few days ago. These are the pathways, and they're occurring over time. So the x-axis is time, and the y-axis is a numerical value, which represents whether that pathway is elevated or not. The dotted line is where the normal controls would sit. So you can see that all of these are elevated. Some are coming down over time, like the interferons and the reactive oxygen species, but some of them are persistently elevated above normal controls. These are the pathways which we feel are the most important. Just to give you some perspective of what's upstream, what's downstream, this is very simplified, and all the biologists in the crowd know this, very simplified. There's all sorts of other interactions and feedback going on, but we wanted just to create a model that was somewhat palatable. And in general, your interferons, your TNF are upstream, and then it works its way down. So with our drug repurposing, we found two drugs, upadacitinib, which is a JAK1 inhibitor, and it strongly inhibits the IL-2, IL-6 pathways, and moderately inhibits the interferon pathways, and is somewhat effective against TNF. So we feel with this gives us a lot of upstream inhibition, which hopefully will influence downstream effects. Just for a visualization of the JAK inhibitors, there's quite a few of them. We worked with upadacitinib because it seems to be more specific for the particular receptors we're interested in, and it also has a relatively good safety profile. And it meets, again, the interferons, the TNF, the IL-6, the IL-2. It also affects TGF-beta. Here it is again with our next drug, which is pirfenidone. Pirfenidone is very effective against IL-6 and TNF. It's reasonably effective against the interferons, and it's also reasonably effective against reactive oxygen species. So again, we've got quite a bit of upstream activity, but we also have some downstream activity. A schematic, again, just showing some of these actions. In the upper left, you've got its anti-inflammatory actions. On the right, you've got its anti-reactive oxygen species actions. This is a drug which is used in interstitial fibrosis of the lung. Now, with those two drugs, and with our biomarkers, we've moved into our phase 3 double-blinded, placebo-controlled, multi-arm platform trial. We're enrolling participants from Brazil, Canada, Italy, Uganda, Zambia, and USA. So we're on 4 continents, and we're at 8 sites. We have a focus on low and middle income countries. So it was also important to us as we were doing our drug repurposing that we had a drug that could be taken orally and that would be stable for transport and storage in these various areas. Lots of biologics came up in our drug repurposing, but those biologics were very unlikely to get to those countries or to lower income individuals. If you want to read about this, it is on clinicaltrials.gov. And we chose to take a platform trial approach, and if you recall my discussion from a few days ago, a platform trial is an adaptive trial. These came around during COVID when there was a need to move quickly. So, your traditional drug trials we talked about is usually a drug versus a placebo. You have a sample size calculation. You either meet your goals or you don't, and you may do some post hoc analyses. But in general, it's a very rigid way of approaching the problem. And if you're wrong and you have to go back with another drug, you're starting almost right from the beginning. With a platform trial, you have one big umbrella, and you've got a lot of capability underneath to change as you go, to adapt. So you can start off with multiple arms. In our case, we have 2 drugs, and we have 2 placebos. We have 2 placebos because the dosing of the drugs is different. And there's some other differences as well in terms of how we've over-encapsulated, so they each have to have their own placebo. We're going to enroll a certain number of patients based on a sample size calculation, at which point we do an interim analysis. And at that interim analysis, that's when we have to make decisions, and we have rules all the way through in how those decisions are made. We can drop both drugs if they're futile. We can continue with one or both drugs. We can do add-on drugs. We can add another drug on altogether. As we approach this, we will submit the data back to the regulatory agencies, and we'll be doing amendments on how we want to move forward. But what it does is give it an incredible amount of flexibility, and you can move much, much quicker. So what exactly are we doing? We have our primary objective is to assess the efficacy of repurposed drugs compared to their respective placebos in reducing standardized symptom severity scores in participants with long COVID. For an outcome measure, we're using a validated measure from Birmingham called the Symptom Burden Questionnaire. And we're looking in 5 areas: fatigue, breathing, circulation, neurologic, and myalgia, arthralgia. We also have a number of secondary objectives which are related to safety, but also to other symptom burdens such as anxiety, depression, and overall well-being. We chose these 3 scales because we used these 3 scales in our last study, and there were significant differences where those with long COVID had higher levels of anxiety, depression, and lower quality of life. We're also looking at exercise ability, and then we've got our biomarkers. For our biomarkers, we're doing pathway-specific proteins, so those TNF, the IL-2, the IL-6, the interference, and some of their downstream proteins, but then we're also using the long COVID-specific proteins, which I already described to you. And we have rules on how we approach the analysis of the blood biomarkers. And then we're fortunate to be able to do a comprehensive omics examination, and that would go right from genotyping all the way up to proteomics. So if we have a patient in our post-hoc analyses which doesn't respond to a drug, we want to know if there's a biological basis as to why they didn't. Do they have a variant? They were never going to respond to that drug. Did that drug not get well absorbed so it didn't have the actions and so forth that we expect? So we'll have some tremendous data there, and of course we'll be happy to share it and work with people in the room. Our inclusion criteria, we're working with adults age 18 to 65 years. They have to have a previous COVID-19 infection, which includes a positive test for either nasopharyngeal PCR or an antigen test. If they don't have one, then we draw blood and we do N-capsid antibodies. And at least early in the pandemic, up to 80% of patients would have N-capsid antibodies for at least 8 months. So we're really trying to identify those patients who have had a SARS-CoV-2 infection to be really specific on what we're looking at. We're using the World Health Organization definition, so 3 months out, they have to have continuous or new symptoms, and then they have to be experiencing one of the following: fatigue, breathing, circulation, neurologic, or muscles or joints. So how did we get to those outcomes? These 5 symptom clusters were selected based on unpublished data from the National Institutes of Health and Care Research in the UK. And they had a good alignment with the 5 validated SBQ scores. The selection was driven by their prevalence, and this was in a second validation cohort that was very random, coming to their primary caregivers. So it had to be prevalent, and it had to have impact on their quality of life. That's how we came up at those 5 clusters. They have to be willing to participate throughout the study, and they have to be able to provide informed consent. I won't get into the exclusion criteria. There's way too many. Both of the drugs we're working with are immunosuppressants. And when you're working with immunosuppressant, there's a tremendous amount of organ work you have to do, as well as infectious disease work. I won't get into that, but when a patient comes in, they'll be consented. They'll be educated on the process. They'll start off with their surveys. They have 1 week, because some of them will have difficulty with all the surveys. They'll have 1 week to complete them. We'll do the blood work, and we have 1 week to get the blood work back to make sure that they meet all the criteria. What is the SBQ? So the SBQ was developed by Sarah Hughes and colleagues again in Birmingham. They did an initial validation of it, but it was in a very limited, restricted participants. We reached out to them and formed a partnership, and they were absolutely fabulous. I can't say enough about them. They were so interested in participating, and they told us that they had repeated the validation again in a relatively random population that had presented to their primary care physicians. With good results. And they were kind enough to share that data with us, which is all unpublished data. So I can't show it, it's not my data, but without that data, we would not have been able to meet some of the assumptions we needed to for our statistical work. But again, we'll be using 5 of the scales, each of which has very strong psychometric properties. The respondents rate their symptom burden over the past 7 days using a dichotomous or a 4-point rating scale. Each scale covers a different domain, generating a summed raw score that is then converted to a transformed score of 0 to 100, with higher scores indicating a greater symptom burden. So we did use a fairly traditional sample size calculation based on this information, and using this unpublished data, we were able to calculate the required sample size on all 5 SBQ scales, and the smallest effect size was the one that we really focused on to make sure we got all 5. And based on this calculation, our phase 1 sample size is going to be 348 patients, or 87 patients per arm, and we feel that would be adequately powered across all 5 scales with the assumption of the parameters as seen. We've included a dropout rate of 25%, and what we're looking for is at least a 50% drop in symptom score. We do our interim analyses. This gets a little bit more complicated. It's a heterogeneous disease, it's a complicated disease, and therefore the analysis is also relatively complicated. We have rules all the way through, which have been well thought out by our statistical team. So at the end of our phase 1, after we have 348 phase 1 completers, we're going to examine the safety, treatment efficacy, and futility. We're using a Bayesian approach, which means as we move forward from phase 1, we can incorporate the data from phase 1 so that it's always moving. It's a very fluid statistical way of looking at the data so that we're always keeping on top of things. And we have reports of means, standard deviations, credible intervals for treatment and control arms. Our efficacy criteria, we're looking for a greater than 80% probability of having a 50% drop in the SBQ scores. If met, the treatment arm continues to phase 2 and the results are kept separate. If 50-80% probability is met, then phase 2 uses a 2-to-1 randomization favoring this arm and the results are combined. Futility, if we have a less than 5% probability of a 50% SBQ score decrease from baseline to control, and if futile, the treatment arm is discontinued and additional treatments are considered. If not futile, we'll continue into phase 2. And again, our sample size is continuously updated at each interim analysis, so we use the current data to update prior distributions and study arm allocation. With that, I think I will stop there. I'm hopeful that we will enroll patients starting in the next few weeks. Almost all of the sites have the regulatory approval. The only lagging sites are in the European Union, which is a little bit more of a difficult process to work through. Ethics is almost completed at all sites, and we're just going through the process now of site visits, doing education, audits, and so forth. We have hired a CRO to work with the drugs. That's one thing we didn't feel comfortable with, so we have a company that sources the drugs, they procure the drugs, they do the over-encapsulation of the drugs, they do all the labeling, and they do the distribution and storage. So we're very thankful to have them involved. Makes things quite a bit easier. As you can imagine, we're working in multiple languages. We've got French, English, Spanish, Portuguese, Brazilian Portuguese, Italian, some interesting languages going on in Uganda, and so forth. So there was a lot of translation and certification we've had to do over the past few months, but that's all prepared as well. And our treatment time is one thing I haven't discussed. We're treating for 3 months, and then we're following patients for an additional 3 months. So the entire enrollment phase for an individual patient will be 6 months. What else can I tell you? I think the only other interesting aspect is we've also negotiated with the CRO. If the trial is positive, those that receive the placebo will also get the drug, which we feel is just the right thing to do. So with that, I will stop. And we are at Western University in London, Ontario, Canada. We are supported so well by members of SILC, which is the Schmidt Initiative for Long COVID, and it's tremendous to have 3 other members here. We've got John, Colin, and Adam. I believe Victoria's back in her room, but it's fantastic to have you guys here there she is, for the first time. To have you guys with us, first time at an independent conference. So this is absolutely wonderful. This is only part of the team. I'm shameful that I don't have a picture of everybody, but we are so fortunate to have an absolute wonderful team. And you can see John in the back, right in the middle with the dark glasses on, looking like the leader. So thank you very much, and appreciate everything everyone has done for us.